like to welcome Dr. Ketan again, please.
Thank you so much. First of all, I would like to apologize. I don't know what happened. This is the first time. Thanks, Amit and Ron, for this invitation. I'd like to share some recent work touching upon cell-cell communication. So I will try to go fast, given that we lost 15 minutes. Okay, again, apologize again. Okay, you can see my slides, right?
Yes, slides are great. Thank you.
Okay, great. Let's go. Oops. Okay, so as Amit mentioned, so we have some experimental work going on to try to understand the spatial omics feature of heart and bone marrow and pediatric cancer. Those are still work ongoing. So today, instead, I thought I would like to describe two computational methods that hopefully can be useful for understanding cell-cell communication based on single-cell analysis. The first one is called CytoTalk to infer signal transduction networks using single-cell RNA-seq data. The second one is a general framework to integrate single-cell omics and imaging data. I'll go over both methods. So in my opinion, I think one of the critical goals of spatial omics is really to understand signal transduction between cells. As you know, it's very complicated. For instance, in this example, tumor microenvironment signaling, as you can see, it's very complicated, involving many different cell types. In fact, we could even think of it nowadays as a signal transduction network, right? So needless to say, understanding such signal transduction pathway is very important for really understand development and disease, not just cancer. Historically, many methods have been developed to infer signal transduction pathways using bulk data, but they suffer two complications from computational point of view. The first one is so-called, as you probably all know, curse of dimensionality. Here, what that means is the number of genes you want to study far exceeds the number of independent measurement you can obtain, be it number of patient samples or conditions. On top of this complication, as we all know now, the gene expression measurement is a convoluted measurement of many cell types in the tissue we measure. So thankfully, nowadays we have single cell data, which hugely alleviate both complications. Because for each single gene, we can have nowadays hundreds to thousands of independent measurements for a given cell type, which makes the computational modeling quite interesting, and which I will show you. So taking advantage of a single cell data, several groups have actually developed methods to infer signaling interactions using single cell RNA-seq data. Several representative methods shown here. Those are great advancement, but I think one missed opportunity here is these methods mostly only look at ligand receptor pairs. They're trying to identify, as shown in this cartoon, they're trying to identify cell type specific ligand receptor gene expression pairs. So as we all know, the signal transduction pathway is not just the ligand receptor pairs. This is illustrated by this one particular example, which involves the fibroblast receptor, fibroblast growth factor receptor signaling. So for the FGFR signaling, there are actually from the same ligand receptor upstream, there could be 4 different downstream pathways. These are the 4 canonical pathways people have figured out over the years. One of them is the JAK-STAT, the other one's protein kinase C pathway. Third one is the RAS and MAP kinase pathway, and finally the PI3 AKT pathway. So depending on the cell type or the condition, different one can be activated. So this is further illustrated by using this single cell data on mouse mammary gland. So what we're looking at here is fibroblast on the left and the luminal epithelial cell on the right. So here we computed the cell type specific gene expression for the different pathway genes for the 4 branches, as you can see, JAK-STAT and so on. For this particular cell type pairs, actually you can see mostly it is the PI3 kinase AKT pathway that is active in this 2 type of cells. So this is further quantified in this bar plots. I can see this is the most active pathway here for this particular pair. In contrast, so if we use another single cell RNA-seq data set, this is again, this is mouse skin cell, I think. Note here, this is the same ligand receptor pair, but again, in this case, it is the JAK-STAT pathway that downstream of the receptor that is the most active. So, this example basically emphasize the importance of looking at the entire pathway in order to understand their behavior. So towards this goal, very talented graduate student, former graduate student in my lab, Yuxuan Hu, developed this algorithm called CytoTalk. So this is recently published. So the basic idea is we want to treat, we want to infer the entire pathway de novo. Okay, so we treat this problem as finding a communication network between 2 cell type problem. So in the next few slides, I will give you just a little bit under the hood description of the algorithm. Okay, so at the core of the algorithm is to rely on this price collecting Steiner tree problem. So this is a classic graph theory problem as shown in this cartoon. So basically in this problem, you have nodes and edges, both are weighted. So the node weight represent price and edge weight represent cost. So the algorithm try to seek a subgraph that maximize the sum of price and same time minimize the cost of collecting those price. So what does that mean in our problem? So we can draw an analogy here. So in our problem, we also have node price and edge cost. So the nodes are genes, edge represent interactions between genes. We can construct this integrated network using single cell RNA-seq data. So let me just explain a little bit more. So here, the node price contains 2 kinds of information. One is the gene expression specificity of gene Ka in one cell type A. It also contains information how closely connected that gene is to the receptor gene. Okay, because we want to infer signal transduction pathways. So that is node price. And for edge cost, basically we use a measurement of loss of expression correlation between 2 genes. For our work, we use mutual information between 2 genes, so shown here. So with this problem set up, so we apply the PCST problem, then that will give us a optimal subgraph from the integrated network that represent genes that highly specifically express to the cell type involved, and also closely connected to the ligand receptor genes. Here we also require there's a strong crosstalk score between the receptor and ligand genes. Basically, that requires highly expressed gene and also very specific expression between for the ligand receptor gene. So once we have this optimal subgraph, that is the output of this algorithm. Okay, any questions so far? Okay, I will keep going. So as I mentioned, it is actually an open question. How do you evaluate the prediction of those signal transduction pathways, because the biggest problem is the lack of gold standard. So in this study, we developed two approaches to evaluate the performance or the quality of the predicted pathway. This cartoon shows the rationale for our first approach. So we used spatial transcriptomics data such as SIG-FISH. Here, our rationale is if a pathway is true, we would expect the genes in the pathway have a high correlation if those are from cells that are physically close to each other. Otherwise, the correlation will be very low or almost close to random. So we can assess for a given pathway by comparing these two distributions, we can get a sense of whether the prediction has some kind of support or not based on the SIG-FISH data, which is orthogonal to our single-cell RNA-seq data. So for SIG-FISH data, we use this particular data set published by Long Kai's group recently. So this is on mouse visual cortex. It covers a number of cell types such as neuron, microglia, endothelium cell, and so on, as you can see here. We choose this data set because there's a matched, same tissue, matched single-cell RNA-seq data as shown here. Okay. For these cell types that are covered in this tissue, several of them are known to interact based on years of research, which is summarized in this review. For instance, astrocyte is known to interact with endothelium cell, and a neuron is known to interact with astrocyte, which is well known. And also microglia is known to interact with endothelium cell, and so on. So we applied cytotalk to the single-cell RNA-seq data only. So this is an example of predicted signaling pathways. So the green edge here indicate ligand receptor interaction. The blue and yellow edges represent intracellular interaction networks, as shown here. Okay, and the node size represents absolute expression level of a gene, and the node shade color represents expression specificity for a given gene to that cell type. Okay, so using that metric based on physical distance measure, so we compare cytotalk to several other published methods. These four methods, CellPhoneDB, Kuma, Scaly, and Zhou et al., only predict ligand receptor gene pairs. These two other methods, NicheNet, SOPTSC, predict the entire pathway, but only based on known pathway, so they are not de novo reconstruction. So what you are seeing here on the y-axis is the p-value for the pathway gene expression correlation difference. So that's a long word between close cell pairs versus distant cell pairs. So the SIG-FISH data give us actually the spatial distance between any pair of cells, so we can calculate that. So here, as you can see, the correlation of the predicted gene pathway is much higher for cytotalk compared to the other methods. So here, each dot represents the entire set of a predicted pathway for a given cell type pairs, as shown here. So, so each box plot has 8 dots here. Okay. So this is the evaluation using the visual cortex data. We also evaluate the performance using a similar matched data set on olfactory bulb. Here, again, you can see the correlation of close gene pair, cell pairs much higher for the pathway predicted by cytotalk. Okay, so we also tried a different way to a second approach to evaluate the prediction, which is we try to find single-cell RNA-seq data with the receptor gene knockout. So fortunately, we found this this particular publication by the Ahmet group, where they identify a novel interaction between the AT2 cells and the basophils in lung. And that's mediated by the interleukin-33 ligand and interleukin-1 receptor, like 1 receptor interaction as shown here. So they validated their finding by knocking out interleukin-1 RL receptor and did the RNA-seq, single-cell RNA-seq profiling. So we took advantage of that data set, and then we asked, okay, among the predicted pathway genes, how many of those are differentially expressed? Because our rationale is if the pathway gene is true, then we would expect differential expression if you knock out the receptor gene protein. So, so using that data set, we construct a ROC curve here, shown here, compared to the 2 other methods because they predict the entire pathway. So here, the true positives are the genes that are differentially expressed between the receptor knockout and wild type cells. As you can see here, the AUC value is significantly higher for the cytotoxic prediction compared to other 2 methods. This is another way to look at the result. The pathway gene predicted overall has a significantly higher differential expression p-value for the cytotoxic prediction compared to the other 2 methods. Okay, so taken together, these evaluations suggest that cytotoxic can have a pretty accurate prediction, at least improvement to existing methods. Then we apply cytotoxic to perform a pan tissue analysis for signal transduction pathways. We are particularly interested in this phenomenon of signal transduction pathway rewiring. Okay, basically the connectivity of the pathway genes are different between different conditions. So in this case, we took advantage of the recently published Human Cell Atlas published by the Guo lab in China. So we identified the 3 tissues that have matched data between fetal and adult stages because we are interested in the developmental aspect of the rewiring. And we focus on 2 particular cell types. One is endothelium cell, the other one is macrophage. Both cell types are very important. As you know, endothelium cell line the entire interior surface of the circulation system. And macrophage is a key player of the innate immunity, and both cell types are known to interact. So this 2 Venn diagram shows the number of predicted pathways for the 3 organs between these 2 cell types. So there is about 50% overlap of the pathway between the different organs across both developmental stages. So suggesting there's quite a bit difference in terms of signal transductions for the same cell pairs in the different organs. So we further examine this issue. So again, remember we want to identify a rewiring event for the gene in the network, in the pathways. So we calculate this metric called the signaling entropy shown here. So for instance, for this node here in this predicted pathway, we can calculate its entropy, signaling entropy using this formula. Here, Wij is the weight between gene I and gene J, as I described in the previous slides. So what this signaling entropy measure is essentially the uncertainty of the interaction pattern of a given gene with other gene in the pathway. Okay, basically, other way to say it, it's the likelihood of this gene will rewire in a different tissue. So we compute this signaling entropy for all genes in our predicted pathways in both fetal and adult organs. Surprisingly, we found actually the signaling entropy for the adult pathway genes are significantly higher compared to the fetal pathway genes as shown here. So this suggests that the adult signaling pathway may be more plastic compared to the fetal version, even for the same type of cells. Okay, so overall, these analyses suggest there's a significant amount of heterogeneity in the signal transduction pathways, even for the same cell types across different tissues. Maybe that's a strategy for the cell type to gain tissue adaptation in different tissues. Okay, so we also find that these pathway with those dynamic rewiring nodes tend to be enriched for tissue-specific functions or diseases shown here. Each column here is the sentinel rewiring node for that pathway. So you have three tissue types and both fetal and adult stages. You can see for the kidney genes, they are involved mostly in the kidney function or disease and so on. So any questions so far? Okay, I will move on. So I will switch gear a little bit to talk about GLUR, which is the method we developed to integrate single cell omics and imaging data. There's a lot of challenges in integrating single cell data, which some of those I shown here. First of all, as we all know, single cell assays for a single modality have been developed nowadays. They are getting more and more robust. We can use those assays to measure different molecular analytes, such as DNA mutation, transcriptome, epigenome, and even 3D genome organization. But all these assays only provide a partial view of the cell. On the other hand, true multi-omics single cell assays are still in the early stage of development. Spatial omics data is great, but typically the number of features that can be captured is much smaller than non-spatial omics data. So this leads to a lower resolution to resolve cell types or cell states and so on. And a related question for this issue is there's a lack of standards for evaluating the performance of the different integration methods. I think as a community, these standards need to be developed as well. Okay, so a number of groups are working on this problem. So we also worked on this problem recently. We developed this GLUR algorithm. This is spearheaded by a very talented postdoc, Pentao, in my lab. So this is we think this is a general framework to integrate single cell multi-omics data with imaging data, as shown in this cartoon. So in the next few slides, I'd like to just give you a brief overview of the major steps of the algorithm. So the first step of the algorithm is really to do a joint dimensionality reduction of the different data modality. Here, for each data modality, the data can be represented as a matrix. So we use this joint non-negative matrix factorization, a well-known algorithm to do this. So the result of this step is a set of shared latent features for the multimodality data, as shown here. And then using, we try to match the cells from different modality in this latent feature space, as shown here. For instance, this could be a single cell RNA-seq data. This could be ATAC-seq data. So we use the mutual K nearest neighbor algorithm to do this, but in the latent feature space. Okay, and then finally, So in GLUR, one data modality is treated as a reference modality. For instance, it could be single-cell RNA-seq data. The rest of the modalities that are the query modality. And then we're trying to find a mapping function between the reference latent features and the query latent features. So we do this using a convolutional neural network because it can handle nonlinear functional relationship. So after this step, basically what we have is these set of learned functions that can map the latent features from one modality to the reference modality. With these learned functions, we can then finally do a co-embedding of the different data modality. And with this co-embedded data, we can do a number of other downstream analysis, such as imputing both for the single-cell omics data and the imaging data and other tasks, for instance, signal transduction pathway inference. Okay, so how do we evaluate the integration accuracy? As I mentioned, this is an open question. So here's what we came up with. First of all, we need to define the ground truth for the cell-cell pairing from different modality. In this exercise, we took advantage of true dual omics data set, such as dual omics data that measures both RNA and ATAC in the same single cell. And then you need the performance matrix, right? So we consider three types of metrics. The first one is the degree of mixing of cell of different modality in the co-embedded data. Secondly, we can consider the degree of separation of cell types or states in the co-embedded data. And finally, we devised this pretty simple global metric we call integration accuracy. Essentially, it's the percentage of true cell pairs because we have the ground truth among the predicted neighbors of a cell. Okay. So how does GLUR perform? First, we use the true duomics data set generated using the SCRNA-Seg, a protocol developed by Kun Zhang's group at UCSD. In this particular data set, they profile a mixture of four cell lines, BJ, H1, K562, and GM12878. So what you see here is the integration result for the three methods, Surratt and Liger. I'm sure you all are very well aware of these two pioneering methods. So the cells are visualized based on the modality. So in terms of the first metric, a mixing of different modality, Surratt and GLUR does a very good job, but Liger seems to have some problem with well-mixed cell. Okay, and then if you look at the degree of the cell type separation, in this case, because of cell line, we know for sure what is the ground truth. Again, you can see both Surratt and GLUR have very nice cell type separation, but there are some mixing for the Liger result. Okay, so the cell type identity can be confirmed using the known cell line marker genes as shown here. This is the original RNA-seq data. This is the co-embedded RNA-seq data. As you can see, it's not that clear, but you can see the cell type marker gene is very well separated for the GLUR prediction compared to the other two. And again, using this integration accuracy metric, we show the accuracy is significantly higher for the GLUR integration result. So that is using cell line mixture, which is a relatively simple, easier problem. Then we use another SCRNA-Seg data set on primary tissue. In this case, it's the mouse cerebral cortex data. Okay. So again, you can see first looking at the data modality mixing. This time, Surratt seems to have some problem mixing the data. Liger have very thorough mixing of the different modality, but in terms of the structure in the data, it's not very clear. By structure, I mean clusters, right? So if you look at gluer, there are some clear cluster here, and those clusters actually represent the known cell types in the cortex. So this is confirmed by the marker gene for these different cell types and shown here. And more importantly, the corresponding ataxic data signal is concordant for the same cell type for those marker genes as shown here. Okay, and finally, the integration accuracy is significantly higher for gluer. So for the rest of my talk, I'm just gonna show you some, you can think of it as a utility of gluer, but also we can sort of measure the integration accuracy between single cell RNA-seq data and the multiplex imaging data. I'm sure many of you are familiar with this CODEX protocol developed by Gary Nolan's group at Stanford. Essentially, it's a multiplex imaging protocol that can measure up to 50 to 60 protein markers at single cell resolution, shown in this cartoon. So here, this is the ongoing experiment in my lab. We are looking at bone marrow using a panel of 11 protein markers using CODEX. So to evaluate the integration utility, we use the original CODEX publication here by the Nolan group. Here they profile 30 protein markers for mouse spleen. And there is also matched single-cell RNA-seq data. So first of all, if you look at the original data without integration, you can clearly see clusters in the single-cell RNA-seq data. But it is not that clear for the CODEX protein expression data, probably due to the much smaller number of features in the CODEX data. And after integration by gluer, we can clearly see clusters for both the co-embedded RNA-seq data and the protein data. And these different clusters represent known cell types in the mouse spleen. And the different clusters clearly show cell type specific gene RNA expression and the protein expression. This is an example of the VCAM1 gene. It's a marker gene for the red pulp macrophage in the spleen. So you can see the expression level for both RNA and protein after data integration is very specific to cluster number 12, which is the red pulp macrophage. And this is another example, NCR1 gene. So it's the marker gene for the NK cell. In this case, you can see again a clear, very specific expression in cluster number 16. Okay, and then we also show that gluer can be used to integrate single-cell RNA-seq data with seq-fish data, which is a high-throughput single-cell transcriptomics data. In this case, we use the mouse olfactory bulb data, which I mentioned briefly before. Here, again, if you look at the original data without integration, you do see some clusters in the seq-fish data, but the separation is not as strong as the RNA-seq data. Again, likely due to the lower number of features in the data. And after data integration, we can see many clusters representing the different cell types in the olfactory bulb. More importantly, cell type, the same cell type, but from different data modality clustered together. For instance, here, so here S post-fixed X means cells from the seq-fish data, and post-fixed NS means cell from RNA-seq data. For instance, here you can see the endothelium cell from both modality clustered together, the oligodendrocytes from those modality clustered together, and so on. Okay, so I hope I give you a rough overview of the two algorithms we recently developed. Just to summarize, I have argued that just by looking at ligand receptor pairs is insufficient to understand cell type specific signaling pathways. We therefore developed the cytotalk algorithm for de novo inference of full signaling pathways using single cell transcriptomics data. We used any single cell RNA-seq data, but presumably single cell fish data could also be used moving forward. We also developed this GLUER algorithm for, it's a general framework for integrating single cell multi-omics data with imaging data. So we have evaluated the performance of GLUER using a diverse set of data sets. I show you some of those. Based on our evaluation, we showed there's a improved integration accuracy by GLUER. With that, I would like to thank people in my lab, particularly Tao Peng and Yuxuan, who spearheaded for the development of these two algorithms with help with Greg Chen and other lab members, and also collaborators and funding resource. Thank you for your interest. I'm sorry again for the glitch at the beginning. Be happy to take questions.
Thank you, Dr. Tan, for this fantastic talk. So we can take questions, and you might want to either post on the chat or unmute yourself and feel free to ask directly. And in the meantime, we have one question from Chen Wang. What's the ground truth to train the model that integrate the codex and single cell RNA-seq?
Right. That's a good question. There is no ground truth besides the known cell types people know. So the model, when you say the model training, you're probably referring to the convolutional neural network. We use actually the convolutional autoencoder, which is the unsupervised method, because we mainly want to map the function. We want to estimate the function mapping the latent factor between different data modality.
And any other questions? As we wait, I have one question about the signaling cytotalk, right? So the measurements are coming from RNA-seq, but signaling happens in the protein level, and it has phosphorylation and non-phosphorylation states, right? So how do these agree, or what is the concordance? Would you comment on it?
Yeah, that's 5,000 mile question. Yeah, I mean, clearly using RNA-seq data is suboptimal because we know, as you mentioned, post-transcriptional regulation, post-translational modification. But that's all we have now. I know it will be better if we have single cell proteomics data, but that's probably far away. Well, not that far, but still. But we do see, based on if you take the predicted pathway by cytotalk, we do see enrichment of known pathway based on pathway database such as Reacton or pathway common. So I guess that's one good sign.
Mm-hmm.
And we use experimental data, for instance, by looking at the receptor gene knockout, we do see there's a good agreement of the pathway gene and gene that are differentially expressed. So obviously, some of those differentially expressed genes may not be direct pathway, maybe further downstream, but yeah. Okay. I totally agree. I think generally there is a lack of reference for this issue.
Okay, thank you. And there's also a question from Hua Li. What is the source of ligand receptor interaction pairs provided in cytotalk?
Right, so you might know there is a ligand receptor database developed by the Forest Group, published many, I don't remember, 5 years ago in Nature Communication. We took that database and supplemented with additional literature search. So we add about maybe, I don't remember exactly, several tens of additional ligand receptor pairs.
Good, thank you. Any other questions? And as we wait, the glue actually is fantastic. You kind of moved the one map, imaging map, to a nice combined map, right?
Yeah.
So my question is, the RNA-seq and imaging analysis, they have different technical noise and they have different detection sensitivity. So do you take into account in your model these differences? I mean, learning probably learns it, right? Machine learning, but is there any other like normalization factor to it?
Yeah, that's a great question. We just, well, we didn't expressly consider the noise or the feature of the different data modality. We just use standard, you know, pre-processing or normalization techniques for the different data modality. For instance, a single-cell RNA-seq data, we can normalize the batch effect and so on. The codex data, we just rely on the existing pipeline for codex data analysis to remove some noisy cells, for instance.
I see. Sounds good. And any other question? If not, let's thank to the speaker again. I'm going to use this reaction called clapping, and thank you so much. And of course, we don't see a lot of cameras, but I'm sure people are you know, clapping as well. And as I mentioned, Dr. Fan is in an NIH review panel today, so he couldn't make it. And sorry about that. But I think hopefully this will be very useful for our community, the way that signaling was modeled. Thank you and your team's efforts. Thank you so much.
Thank you so much, Ahmet. Again, apologize for the glitch.
No worries, technical things happen. Please go ahead.
No, I was going to say have a good weekend.
Have a good weekend, everyone. Before we conclude, I'd like to also mention that next week, 3/19, we have Dr. Seng Zong from UCSD talking about natural display of nuclear encoded RNA on the cell surface and its impact on cell interaction. And then I hope to see you guys next week. And again, thank you, everyone, and have a nice weekend. Bye-bye.
Bye.
Thank you, Yanshin, for the help as well.
Yeah, no problem.